Respected chairperson, senior doctors, and my dear friends, at the outset, I thank the organizers, Krishna Seshadri and Vijay Kumar, for this opportunity. And we all know beta cell are the insulin secreting cells in the pancreas. And in type 1 diabetes, there is destruction of the beta cells by the immune system, by the autoimmunity. And in type 2 diabetes, there is gradual worsening in function of the beta cell, and whatever beta cell is not very efficiently functioning. And as you all know, during the diagnosis of type 2 diabetes, 80% of the beta cell function is lost. Whether it is type 1 or type 2, there has been a loss in beta cell function, which has been the disease process. So conquering disease or conquering diabetes may not be possible without building the beta cells. Let us try to discuss the possibilities by which we will be able to build up the beta cells. I would start my talk by recalling the Bantic lecture during the invention of insulin, and you can see the Nobel lecture which he delivered in Stockholm. Insulin is not a cure for diabetes, it is just a treatment. The same thing has been confirmed by various epidemiological studies, and DCCT, one of the major landmark studies in type 1 diabetes, and the follow-up EDIC study, which has very clearly shown intensive insulin treatment incontrovertibly reduces the risks of retinopathy, nephropathy, and neuropathy that is associated with type 1 diabetes. However, it does not fully replace pancreatic beta cell function. So unless you are able to replace the pancreatic beta cell function, it is very, very difficult for us to get the complete cure of diabetes or to conquer the management issues of diabetes. Now, what could be the real possibilities of building up the beta cell? One, you can have a pancreas transplantation where you have various types of pancreas transplantation. You can have a pancreas kidney transplantation, pancreas after kidney transplantation, pancreatic transplantation alone, where you try to totally replace or replant a pancreatic beta cell. Of course, you have islet cell transplantation. It has to be noted, like our pancreas weighs around 70 grams. Out of that, we have around 10 lakh islet cells, and each islet is composed of 1000 to 1500 cells, and 80% of those are beta cells. So transplanting the islet cell is definitely possible. And beta cells from stem cell, this is the most promising way of building up the beta cells. And of course, you can have genetically modified cells which can produce insulin, but they may not have a direct replacing of beta cells. Regeneration of beta cells, as of now, we don't have any drug which totally regenerates a beta cell. Of course, you have claims with gliptin and various other drugs which could regenerate, but these are not really building up the beta cells as of today. So I would focus more on the beta cell from stem cell, how exactly we are able to build up the beta cells. Islet cell transplantation, one of the commonly available beta cell replacement, is that it is theoretically very simple. You take a donor pancreas, you isolate the islets, and these islets are prepared like a blood bag unit, and then you transplant it. So what happens, the recipient, the islets are delivered through the portal system where it dislodges in the hepatic sinusoids and it starts secreting insulin. The advantage of islet cell transplant or the natural regeneration or building of beta cell is that you are secreting insulin in the portal circulation, which can mimic your natural physiology of any insulin secretion. Now, whatever newer drugs we have, newer insulin we have, or insulin pump or artificial pancreas, you are injecting the insulin in the systemic circulation. Normally, your portal insulin concentration is much higher than your systemic insulin concentration. It is very difficult for the artificial pancreas to mimic your normal porto-systemic insulin concentration, which can be possible only by these having a natural way of building of the beta cell. So one, you have islet cell transplantation, but the problem with islet cell transplantation is that first major problem is the availability of the donor. You don't have enough donors. Then the next challenge is that isolating the islets from the pancreas. The next challenge is the immunosuppression. That is one reason why islet cell transplant has not really taken off, though islet cell transplantation has been done in many centers. As of now, most commonly done islet cell transplant is for the chronic pancreatitis, where you do auto transplant. For some reason, you are forced to remove your own pancreas. You can isolate islets, inject it back, which means even after pancreatectomy, you may not develop diabetes. The most promising way of building beta cell is the stem cell, where stem cell, all of us are aware of the embryonic stem cell, but we have adult stem cell. You can derive the beta cell from acinar cell of your pancreas. We can have beta cell from pancreatic ductal cells, We can have beta cells from pancreatic sources and non-pancreatic sources beta cells. Now, of course, you have embryonic stem cells, and there is something called therapeutic cloning, which I would discuss today. So, the first thing is developing a beta cell or generating beta cell from the acinar cells. So, acinar cells or acinar cell lineage is the most closely related developmental to pancreatic endocrine tissues. So, it is very, very easy for us to transform the acinar cells to the pancreatic beta cell and differentiate the beta cell. This concept is something called transdifferentiation, where you reprogram the terminally differentiated cell. You have a terminally differentiated acinar cell, you de-differentiate it and then reprogram it to the another differentiated phenotype, which is possible. How exactly they do is, this is a schematic illustration of what exactly is done. You have a acinar cell, just for convenience, all brown thing or proteins which are expressed by the acinar cell, you de-differentiate this acinar cell, which means you see you have proteins which are seen in the de-differentiated cells. Then again you re-differentiate them by using epidermal growth factor and leukemia inhibiting factor. You have various other factors where it can again bring to the beta cell lineage. You can see once when the acinar cells are de-differentiated and then re-differentiated, you can see they express insulin, they express C-peptide and they express other beta cell functions. So, it is definitely possible to de-differentiate and re-differentiate acinar cells to the beta cells. The another way of bringing beta cells apart from acinar cells is the beta cells from pancreatic ductal cells. The ductal cells they have a very high and rich source of stem cells. And there are two mechanisms by which beta cells can grow. One is replication of the pre-existing beta cells. Second, you can have neogenesis or differentiation of new beta cells from progenitor stem cells that are not beta cells. These progenitor cells are present in the pancreatic ductal cells. So, what is the rationale behind this? We know our pancreatic islets or pancreatic beta cell, they increase by at least 20 times after birth. As you grow, your pancreatic beta cells also grow. And we know in type 2 diabetes and also in obesity, where you have severe insulin resistance, you have compensated hyperinsulinemia, which means your beta cells are able to compensate or your beta cells are able to grow. And neogenesis is supposed to be an important factor or important aspect of building of the beta cell in these kinds of patients. The another way of getting beta cell apart from the acinar cells or apart from the ductal cells is that you can have what is called adult cell reprogramming, where you have non-pancreatic cell source to generate beta cells. For example, liver and pancreas are developmentally very closely related. And generally in lower animals, what you call is a hepatopancreas. You don't have separate liver, you don't have separate pancreas. You have both the thing together, and this liver and pancreas are developmentally very closely linked. And what really matters a lot is that the PDX1. PDX1 is a growth factor or a protein which plays a central role in the pancreas development and also in the beta cell function. If suppose in the hepatocytes, you are able to bring in the PDX1 gene, naturally they can get diverted into your beta cell which can secrete insulin. How exactly these things happen? You have, this is a very, very interesting study where you have a adenovirus. This is the rat where hepatocytes are infected by the adenovirus. You can see after they are infected within 2 weeks, you are able to see the ectopic PDX1 expression, and this PDX1 is responsible for the beta cell differentiation. After it is expressed, you can see this endogenous PDX1 at 2 months are seen around the central vein. And remember, these are the rats which have been made diabetic by streptozotocin. Streptozotocin induced diabetic rats, you inject this PDX1 gene through the adenovirus, and you can see after some time there is amyloidosis of streptozotocin induced hyperglycemia. You can see these are the controls, hyperglycemia comes down. And the more important this thing, insulin in liver, which was produced even after 4 months after viral infection, and this PDX1 is also responsible for the beta cell and some extent of the alpha cell, where you are able to also have a glucagon. So you have insulin and glucagon which are persistent even after 4 months. You can see there's a prolonged protection after that. This is streptozotocin, and here the recovery was at least 80%. And more important, this is phenotypic stability of the modified cells. You can say if suppose I make a hepatocyte to secrete or to modify to beta cell, how far they are stable. Studies have very clearly shown they are phenotypically stable, and liver insulin producing cells are resistant to the beta cell specific toxin streptozotocin. So this is very, very much possible. And in humans, what they have done is, you do a liver biopsy, you take your cells in vitro, and you propagate your hepatocytes in vitro, then you infect the PDX1 with a soluble factor treatment, and you have insulin producing cells which are produced in vitro, and the same thing can be implanted to the patient, which means it's your own cell. You need not be worried about the immune rejection. There may not be a rejection, so which is definitely possible, where you will be able to have your hepatocytes which can secrete or which can transform to a beta cell and which can secrete insulin. And bone marrow derived mesenchymal stem cell, this is other non-pancreatic source of becoming a beta cell. This is very promising things. Bone marrow is a source of two cells. One is a hematopoietic stem cell and another is a mesenchymal stem cells. And bone marrow derived mesenchymal stem cells can differentiate into insulin producing cells. Though they are less pluripotent than your embryonic stem cell, they can also differentiate it, and this bone marrow derived cells can augment your endogenous beta cell regeneration. Apart from multiplying into beta cell, they can also regenerate your beta cell, and expression of PDX1 in bone marrow derived mesenchymal stem cells has been used with the lentivirus. The advantage of these mesenchymal stem cells compared to the embryonic stem cell is that these are less tumorogenic compared to the embryonic stem cells. These are the ways by which your mesenchymal stem cells from bone marrow has been used. Your bone marrow derived stem cells can differentiate, as I said before, to the pancreatic stem cell, and they can differentiate to the endothelial cell, and they can send signals and growth factors to build up your beta cell. And in type 1 diabetes, these bone marrow derived mesenchymal stem cell, they can try to transform the cell so that these cells are not destroyed by your autoimmune process. So it can even stop your autoimmune destruction by these mesenchymal stem cells. Embryonic stem cell, this has been the most fascinating and most interesting field of any medicine. And embryonic stem cell, the advantage is that these are constitutively in the proliferative state. They are pluripotent. They have highest differentiation potential to insulin secreting cell. All of us are born from a single cell which has continuously multiplied, and this embryonic stem cell are the one which differentiate into various organs. The advantage is that these are immunologically inert. The concern with the embryonic stem cell is that they have potential to form teratoma from undifferentiated cell, because you may not be able to cut short the multiplication of the embryonic stem cell. Lots of ethical concern has been there, because if you are not removing this embryonic stem cell, this has a potential or probability to become a future embryo and a future child, because when you are taking embryonic stem cell, you are killing a living organism. So there are lots of ethical concern has been with the embryonic stem cell. How exactly is these things are possible? You have blastocyst, especially you take it from in vitro fertilization clinic, and you have the stem cell. You can have a human embryonic, you can have a micro electropipette where you are able to open down this blastocyst and you can aspirate the stem cell. And to remove the stem cell, the blastocyst is open and the stem cells are aspirated through the pipette. Then what you do is, you add various growth factors, you have various chemical cues, which are added in the petri dish with stem cell. Depending on what I want they to differentiate. For example, I want these stem cells to become a pancreatic cell, I can put PDX, I can put PAX, I can use various differentiation of growth factors. If I want them to differentiate into a neuron, I can do that. If I want them to differentiate into muscle cells, it can do it. Theoretically or hypothetically, it sounds very interesting. And different chemicals and molecules are added to the stem cells to make them or become specific types of cells. The concern what we have is, we are grown up and probably we may not have our own embryonic stem cell. So is there any possibility for me to have my own embryo and own embryonic stem cell? And definitely that is also possible, what you call the ethical overlap between human cloning and stem cell. And this is the dream of transplant medicine, or dream of any therapy, is that where you try to use a cloning technology to create a human embryo for deriving a stem cell that would be a perfect match for the patient. For example, you have a cloning. What you call is cloning is nothing but a somatic cell nuclear transfer, where you can have your own replica of what has been formed. If suppose I have my own cell which is cloned and the next baby is formed, it becomes a reproductive cloning, which has lots of ethical issue. Now we are speaking more and more of therapeutic cloning, where once when the embryo is formed, you have the embryo, you remove the nucleus, and you have your own somatic cell which is transformed. Which means whatever cell it is multiplying or differentiating is similar to my genetic material. And once when the embryo is formed, now you open it, whatever this inner cell mass is there or the stem cell which is there is total identical to my cell, because I have implanted my nucleus into it. So which means I have my own embryonic stem cell. Whatever want I can multiply. If I want my bone, I can get it. If I want my pancreas, I can get it. If I want my neurons, I can get it. So it is very, very simple. And probably if these things succeed, we may be able to build up your beta cell. We may be able to build up any organ, whatever you want. I think stem cell depart, we may have it in near future. So probably when you have more and more competition, you may even have to say, today's special is that you have a pancreas and you have a kidney, and when there's too much competition, naturally you have to give some free. So if you buy a fibula, you'll get a tibia free. So this could be a scenario in the future. So we all dream this to happen. So we will be able to build up a beta cell. You will be able to conquer diabetes. Thank you.